So thanks for having me on. This is really, it's quite novel to be live on Facebook, etc. So let's see if I can, and I might just redo that. Share my screen. I've got a presentation with some sounds and some examples of what we did in the ship tour. So let's see if I put this. Can everyone see that? That's all good? Okay, great. So yeah, I just want to tell you a little bit about the marine research program that we managed to work on throughout the whole of the Poletopole ship tour, and a little bit about my role in it and some of our initial preliminary findings. Now let's see, is this going to, okay. So yeah, a bit about me, because you won't know who I am at all. So my background is that I started off as a field ecologist. I used to work on otters, squirrels, pine martens, etc., and then found myself working on marine mammals. And initially I worked on sort of population studies of large whales, so Bryde's whales in the Gulf and Hauraki Gulf, and humpback whales across the South Pacific. And then more latterly for my master's and PhD on molecular ecology of beaked whales and genomics. So currently I work as a consultant to Greenpeace research laboratories, and they're a small group of scientists working within Greenpeace at the University of Exeter and the labs there. And then I also work as a lecturer in ecology at Exeter as well. And something sort of important for me as well is I'm a Shetlander. I grew up on a rock in the middle of the ocean. So I've always lived beside the sea, and I really feel passionate about ocean protection. I also used to live in Piha in New Zealand. So I lived in New Zealand for a long time. My kids were born there, 2 of them. So, you know, I had a vested interest, like most Kiwis, I guess, in protecting our oceans. So that's kind of my background and where I studied and the sort of organizations that I worked for, leading me to work for Greenpeace. And working at the research labs, I've worked on a number of different campaigns, all sorts of different campaigns, in fact, just supporting the campaigners in each of the campaigns. But the Protect the Oceans campaign obviously is really important within my field of research. And so I was really pleased to be asked to work on the Poletopole ship tour, which is a really, really ambitious, logistically challenging and really exciting ship tour right all the way from the Arctic to the Antarctic. And so my remit was really to try and come up with ideas where we could support research throughout the whole of the Poletopole ship tour. And we were lucky to have support from a foundation linked to Greenpeace Germany. And the aim was to focus on science that could deliver us robust data, but also using the existing infrastructure of the ships. Greenpeace is unique in certain ways in that we have these fabulous ocean-going vessels that can work in the open ocean, and with highly dedicated and skilled crew. And we wanted to use this science to help deliver the core Protect the Oceans campaign message, which Ellie mentioned to start with. So after a lot of discussions and showing about ideas with the core teams and the media and the scientists within the research group that I work in, then we decided to focus on top predators in the ocean, in the open ocean, because they are really important indicator species. And we really wanted to help fill some key knowledge gaps about biodiversity in the open ocean. So in those high seas areas where the ships were visiting throughout the Poletopole tour. So we decided to use some cutting edge molecular monitoring for vertebrate diversity, and I'll go into that, acoustic monitoring for whales and dolphins. And like I say, using the ship's infrastructure and enhance the capacity of ship's crew as well. We know that the crews are so highly skilled, but we wanted to see if we could use this science to build upon some research that might extend beyond the Poletopole ship tour. So, I'm a population geneticist. I've worked on a few different species, but I haven't done EDNA monitoring before. So I did a lot of research as to who are the leaders in the field in this work, and also in acoustic monitoring. So we reached out to a number of really fantastic collaborators that are leaders in their field that could help us carry out this work. And so just to give you a bit of an idea about what eDNA monitoring is, environmental DNA monitoring. It's a really new, emerging, non-invasive monitoring technique. And it's really incredibly useful for detecting rare and cryptic species, because you might go to a site in the open ocean and you might not actually see that much because you're out at sea and it could be challenging weather conditions. But you can actually collect some of the water and filter it for DNA. It's also useful for invasive species. And so we think as scientists, we think that it may well be in the future quite a powerful monitoring tool for monitoring biodiversity in the context of environmental change. So we really wanted to trial this in the open ocean. So just to give you an idea, it's my favorite slide. So the sea, we know there's lots of things living in it, and it's essentially a sort of soup of fish and animal life and plants, etc. And within that water, there'll be fragments of DNA floating around from all the different organisms that live there. And so let's just say we have a humpback whale, might well be traveling through the area, and will stop at some point, carry on, and might do a poo, and we'll leave a whole lot of DNA behind, and we'll swim out the area, and we may or may not see that whale. When we come along with our ship or the ribs, generally we would do this work, and we effectively just basically filter the water, and we are then trapping that DNA on a filter membrane, and we then preserve the DNA with some specialist buffer. And we can take that back to the lab and then try and identify what's been traveling through that water. And so back at the lab, and this is a lab that we work with our collaborators in France, Spigen, they will then extract the DNA, and they'll target certain sequences of DNA that are informative of species ID, and they'll tag those with a molecular tag, run it through a high throughput sequencer, and then generate millions of sequences. And then we will then compare those sequences to a reference database with all the online species that's been sequenced before in that area. And that will give us a percentage similarity to it, so we can help identify the origin of that DNA. And there's a whole lot of different quality control steps that will help us identify how closely that DNA matches to the sequences that are in the reference database. So some of the preliminary results that we had, so the first time we trialed this was in the Sargasso Sea, and that was on board the Esperanza. And so the campaign and the crew, the team on board, carried us out for the first time out in the open ocean. And you can kind of see here exactly how far offshore they were working. So, you know, good 1,500 kilometers off the coast of the US. And they were there for a couple of weeks, and they trialed this. We trained up the crew and the campaign team from remotely. And so it was quite a challenge to carry this out in the deep ocean there. But we still managed to identify around 35 different species across this wide area, including spotted dolphins, and open ocean pelagic fish, so things like tuna, mahi-mahi, wahoo, and flying fish, etc. But then also these sort of mid-water mesopelagic fish, and interestingly, deep sea fish as well. So this pink fish here is the cusk eel, which is actually a really deep sea species. So we're essentially beginning to be able to identify organisms that are right through the water column. And that some of those sites were about 4,000 meters deep. So it's a really interesting technique, and we're learning a lot more about what we can use it for through this work. And what we hope to do, and in fact, we have an intern who's working, she's from Duke University, and she's working at the US office in Washington, or she's actually at home, but she's working with the Washington office, she's going to look through all of the photographic evidence for all the species that they collected during the time out in the Sargasso Sea. So she's actually going through all of these photographs and video footage that the campaign team collected at the same time to identify all the other animals that we may or may not have identified through the EDNA work. And that will help us come up with an idea of an estimate of what species live there in that area, because it's quite a remote place. And so this really helps us identify what kind of estimates of biodiversity we can make with using these three different methods. So the next place that we trialed this method was at the Vemma Seamount, which is about 650 nautical miles off the coast of South Africa. And it's a really interesting seamount. It's right out in the high seas. It has been the focus of fishing previously. And so we were interested to find out what might be living there now, having now been given a little bit of time to recover. And so same thing again, we trialed this EDNA method right out at sea using the ribs. And we found about 41 species, and interestingly, some quite elusive marine mammals. So we found the dwarf sperm whale, which is a really curious, very strange, weird whale. We also found sei whales and humpback sei whales, blue sharks, things like these pelagic fish again, like kingfish, tuna, warahu, and also again the signal for some deep sea fish like these lanternfish. And again, we're going to combine these EDNA data with other streams of data that the scientists and the team on board collected. So things like they had bottom-mounted sound traps for listening to the seamount noises, the whole soundscape for that particular 2 weeks when they were working there, and then some of the images from the dives. And seamounts are really interesting ecosystems in that we know that some can be really very rich in biodiversity, but by their nature, they can be quite difficult to access and research, so very difficult to characterize what lives on them. And so Vemma is actually the first seamount in the Atlantic to be studied using EDNA. And so what we hope to do is, in collaboration with the team at the University of Montpellier, is to combine the data from 3 seamounts right across the ocean. We have Vemma, we have Antigonia, which is south of New Caledonia, and Mont La Perouse off of the coast of Madagascar. And so what we want to do is using this new method is try and understand better what the estimates of biodiversity are on these 3 seamounts. And so we're going to compile all the data together to write a conservation-focused high-impact publication, really using this method to illuminate the diversity of seamounts across the ocean. And so, as Ellie mentioned, I was really lucky enough to be able to join the Arctic sunrise in Antarctic waters, and it really is quite an incredible place to visit, as she will testify to. So here's a picture of Elephant Island, and this tiny, tiny dot you may see in the middle is the Arctic sunrise. It's really quite an incredible marine environment. And so very lucky to work there. And we really did have quite a diverse sampling schedule for our trip there. So we were in the same way, we were focusing on collecting EDNA samples from a number of different locations right across the West Antarctic Peninsula. We were also collecting plankton samples so that we could work with a researcher colleague at University of Exeter to look at plankton health and climate change. We were collecting microfiber samples, so samples of water to identify whether there were microfibers within those areas, and that was following on from work that's been previously done by Greenpeace in previous trips down to the Antarctic Peninsula, and also collecting snow and water samples to look at PFAs, which are they're chemicals that are used in Teflon, for instance, and waterproofing. So this gives you an idea of the sort of areas where we visited, and we were able to collect 103 EDNA samples, including, we actually trialed sampling from the back of the ship on the transit back to Ushuaia, so that no one's done that before, and hopefully we'll manage to get some good samples from that. And after being slowed down through the COVID crisis, where our samples finally arrived at the lab in France in May, so they're currently being extracted, and fingers crossed they're all doing well there. So we really worked quite hard to collect as much data as we could during our time there, because it's such a unique place, and it's also such an amazing opportunity to collect as much data as we can, particularly as the whole of the Antarctic Peninsula is changing quite rapidly as a result of climate change. So EDNA is a bit like ocean forensics. You can imagine it's, it can be a relatively rapid way of estimating biodiversity in certain areas. It can be a challenge, and we managed to sample at the surface throughout the Antarctic Peninsula, but we also managed to sample at depth down to around 300 meters deep using these Niskin samplers off the side of the ship. It tells us what animals may be present in that area. It doesn't necessarily tell us what isn't present, so it's not a perfect technique, but it certainly is something that more and more scientists are using to try and classify what might live in that area. And it may well, like I say, be very useful for monitoring areas that are undergoing environmental change. And throughout our expedition to Antarctica, we also were opportunistically recording all the different whale sightings and whale and dolphin sightings throughout our whole trip. And so we managed to collect over about more than 100 opportunistic whale sightings from 5 different species. And we placed a focus on humpback whale photo ID, because it was something that we could relatively easily do. And we had a fantastic team on board the ship that were able to do this from the ribs. So we identified about 48 different whales, and we don't have to do all of the matching work ourselves by eye. We now can send our beautiful photographs off to Happy Whale and have them matched to online databases that exist as a sort of global catalogue. And we were really lucky to identify 3 different whales that were linked to tropical breeding grounds from 2008 to our trip, 2012, and I think that is 2007 underneath my, So that was really nice to be able to confirm these links to the tropical breeding grounds, and that was in Colombia, Panama, and Ecuador. And let's see if we can get forward. And then we also used a hydrophone system, so a passive acoustic monitoring system off the stern of the Arctic Sunrise. And essentially it meant that we were, from every transit from Amsterdam all the way to Antarctic waters, we were making continuous recordings of whale and dolphin sounds, plus all the man-made sounds that the ship may well have traveled past. So we now have around about 17 terabytes of continuous recordings to go through. And these data were collected by the crew with help from professional passive acoustic monitors. And we're now going through the process of getting someone to go through this data with a fine-tooth comb and extract the most useful parts of that, so that we can use that for other projects. And essentially it sounds an easy thing to do, and it's an easy thing to deploy a 350-meter hydrophone out the stern of the ship, but it takes quite a few people to bring that hydrophone in. So this is kind of what it looked like when we're bringing the hydrophone in from the poop deck. So a lot of hard work. But we were also able to use this equipment to engage some of the supporters and some of the media, and also some of the celebrities that were on board the ship. So this is a very famous actress from South Africa who sailed on the Sunrise out to the Vemma Seamount, Amanda Dupont. And it essentially means that you're kind of giving the ship ears. And so she was able to listen to humpback whales on the voyage and dolphins, and really sort of engage in what the oceans means to her in a way that she probably hasn't done before. So it's really useful to be able to do that and have an extra dimension to the work on board the ship. And so the sorts of things that we're trying to record are things like sperm whales, that are actually apparently the most noisiest animal. And just to give you an idea of what they sound like, let's see if I can get this going. Oops. So that's a sperm whale, actually two sperm whales talking to each other, and these kind of rhythmic clicks. And essentially all whales and dolphins use sound to communicate, which you probably well know, but they are also, they need that sound, and they need a quiet ocean to be able to hear each other. And so the problem of noise pollution is known, and has been known for quite a while. And so what we will, and you can also use these vocalizations to identify different species as well. So what we'll be going through all of these data to pull out the most useful parts of it from our transits, and identify which species were where, and if we're lucky, we'll have enough sperm whale sound so that we can then also use those data in another larger project that our collaborators are working on to help produce an automated or more automated sperm whale detector, which will help people be able to identify sperm whales much more quickly, particularly when they're working around fishing boats. And we also have these small hydrophones that we use from the ribs, and they were really useful in engaging the celebrities that were on board in the Antarctic. And this is two, fairly famous actresses and actors working, listening to the sound of the ice, and just really sort of engaging, again, having this idea that the ocean's not something you just look at, but it's a very busy place under the water as well as above. And I can play you a sound of what the icebergs actually sound like. And that was great in the fact that we were able to, again, engage the media that were on board the ship so that they could listen as well as look at this amazing, incredible habitat that they were working within. So, like I say, this was a lot of work to complete all this science on board the Poletopole ship tour. It really was a collaborative effort in that we were using external collaborators to help us collect the best robust data that we can, and we'll be working with them to analyze this and write up all the publications that will result from this. But we also had lots of internal collaborations, including the highly skilled dedicated crew and the campaigns team, who will be co-authors. And essentially it really does take a floating village to learn about the oceans. There was a lot of people involved in this work, certainly not just me. But it also showed us that the Greenpeace ships are a fantastic platform for conducting research. And we have a lot of data to work on now. So part of my job is to go through all of these data, analyze it, and publish our findings. So, but thanks to all the team that made it happen, and obviously all our funders. And so has anybody got any questions? Shall I stop sharing now?
Thank you, Kirsten. That was awesome. Yeah, I've collected a few questions, so I'll start going through them. And as we said before, if you've got any other questions, just drop them in the chat box now, and we'll hopefully get time. We've got half an hour. So the first one, Kirsten, it sounds like it's quite difficult to do this kind of work in the field. What have you ever had to abandon research because of the actual conditions that you're in, or can you tell us any stories about that?
Yeah, there were some times in the Antarctic where it was beginning to get nasty, the weather, and we did push on, and we were pretty cold, and we pushed on, and when we were working in the ribs, it was, you know, after a couple of hours of working in the Antarctic, even on a good day, you'd get cold. But certainly in a 4-7, we were pushed to finish some of the transects. We would generally try and go back to the ship, have a cup of tea, warm out, and then try and go out again in the afternoon. I think, if you remember right, I think there was only probably once that we actually abandoned it and we waited till the next day. It takes a little bit of time to carry out an EDNA transect, so it's about kind of, you know, half a day for each transect, and we're carrying out 5 of them for each location. So we had a limited time in the Antarctic, and we wanted to make the most of that time, so we really did work in some conditions we probably wouldn't, you know, have chosen to, but you know, always keep erring on the safe side, but generally just getting cold hands. So but I mean, I think hopefully the data will show us that it was worth it. And yeah, so we only abandoned it once, if I remember rightly. And there was one Sunday where we did go out, and it was a bit of a howling gale. We had the third mate driving, and she was, you know, just so excellent that we knew we were safe, and we were cold, and it was windy, and it was snowy, but I like that kind of thing anyway, so we were happy. And we collected our data, so we were all right.
Amazing. The next question is, the DNA samples are a function of place and time, or is this a cumulative sampling along the path?
No, well, it's essentially what we're doing on each of the transects is we're filtering about 30 liters of water, and we're doing that on either side of the rib. So we have 2 samples, so 2 replicates for each of the transects, and we'll do that 5 times. And basically it is a bit of space and time, and so throughout that transect, which I think we worked out to be about 4 nautical miles or something. So any DNA that we manage to collect in that 30 liters will get stuck to the filter. So essentially we won't know where on that transect that particular piece of DNA came from, but we'll know the broad location because we know where we went. It may well tell us the presence of an animal, but it won't tell us what wasn't there. So it's an interesting technique, and it's something you have to slightly get your head around. The other thing that's quite interesting about it is that there are certain species that are probably more likely to be detected using this technique than others. So there are certain species that slough a lot of skin or a lot of mucus, etc. And so it might well be easier to detect those than other species that just don't slough much. So it's an interesting technique, and you have to kind of think about it conceptually, and you're managing to collect DNA in that particular volume of water from something that happens to have moved through it and has left something behind. So yeah, it's quite interesting, and we're certainly learning a lot more about it, and it's nice to be able to work with people that are also very experienced and they've used this technique a lot.
Yeah. And you said, I mean, you mentioned that you've got heaps of data, both from the acoustic monitoring, but also the other stuff that you were taking. What's next for your research? Have you processed a lot of it, or is there still heaps to go through? Where do you go next?
Yeah, well, this is the sort of the stage that we're at now, is we've been really kind of putting together the overview of all the data that we've collected throughout the tour. The EDNA data for the Sargasso Sea and Vemma Seamount, they've actually finished the lab work, and we have the data for that. So like I mentioned in the talk, for Sargasso Sea, we have an intern that's working on all the video footage and all the photographs that they took at the surface and also during these night dives. And so what we're going to do is put all of those streams of data together to be able to write a paper talking about biodiversity in that particular area at that time when the ship was there. The Antarctic samples, those form the largest part of the collection, and we probably won't get the data back from the labs for at least another 4 or 5 months. So that's a large collection of data, and until we get those data back, we have an idea about what we want to do with it, but we need to see what they look like essentially. And then the acoustic data, that is like I say, it's literally continuous recordings, very high resolution acoustic data all the way from Amsterdam to Antarctic waters and back to Ushuaia. And that will take, although it's been run through the first set of computer program that we use, it will take actually a person going through it and pulling out those data and listening for what's the most useful, where the animals were heard, and essentially drawing out the most useful parts of it. And what we hope to do with that, I mentioned sperm whales because they are the most recognizable and in a certain way the easiest to work with of all the cetaceans. And there has been an ongoing project working off of Alaska where they have problems of sperm whales taking fish off the long lines. These fishermen are sort of artisanal fishermen that are working on small boats, and obviously this is a real problem. No one wants sperm whales interfering with fishing. And so what we hope to do is, if we have enough data in our data set, we can then help that project by providing data from another platform where they can then train this sort of automated sperm whale detector, which can then help the whole system work out whether their particular software is working. Because ideally what we'd like to be able to do is help fishermen not have to deal with sperm whales close, and if they know where the sperm whales are, they can not put their lines out. So yeah, I mean that sort of is quite a useful it's a useful sort of application for some of the data that we've collected. So,
okay, awesome.
So lots and lots of work to do.
Yeah, it never ends.
Yeah.
Years after for the scientists.
Yeah.
We've got another question from someone in the Zoom call who asked, it's great to hear about your focus on seamounts and the upcoming report. How does EDNA analysis work with species that have not yet been identified?
Yeah, it's a tricky one. So the way that it works is that when we get the results back from the lab, essentially it will give us almost like a percentage certainty of our result. So there are certain species that have been sequenced and they're in the reference databases, and our sequences that we've collected and generated through our EDNA work will have a really very close percentage similarity. There are other species that have not been sequenced in that particular area, so we might well have a sort of less close percentage similarity, which tells us that it's not exactly the same species, and that might well be the case in quite a few species where you have, and we have these results from the Atlantic, where we get a sort of, it's a kind of a broad match to a family, and we think, okay, well, we think this is a family of fish, or it might be a match to a fish that's been seen or sequenced in the Pacific, and most likely that can kind of indicate that we have a match to a species that is in the Atlantic but just hasn't been sequenced and isn't on the reference database. So you have to sort of tease all of this. This is why we have a really, really very strict quality control criteria from all the way from the lab work all the way down to the bioinformatics, because it's not, not everything in the world has been sequenced. And so we need to know how close and how certain our result is. And this project will hopefully generate more and more sequences over time. And our collaborators hope to set all of this, all of these datas and generate all of these sequences, put it on a sort of global database that will be open access, so that anyone that's sequencing and using eDNA in the future in 10 years time will have more resources to work with than we do now. So essentially we're sort of trying to build upon what people have done before, but also generate more and more data so that people in the future will find it a lot easier than we do, because essentially not everything in the world has been sequenced yet.
Yeah, amazing. And I think you might have already touched on this, Kirsten, in your presentation and some of your answers there, but one of the questions is the eDNA research, is this a new frontier? And specifically they've asked, when the data is collected, will it be churned using machine learning algorithms, or is it going to be processed manually by humans?
Yeah, well, I mean, eDNA or environmental sequencing DNA from the environment is not necessarily new. It's been around for about at least 10, 15 years. But obviously people, you know, it's quite a tricky method, and obviously it's got a lot easier to do with better sequencing technology. Doing it well is quite challenging. And you know, it's done routinely. For instance, in the UK, people looking at great crested newts in ponds will use eDNA tests routinely. So in that sense, in that kind of environment, in, you know, freshwater ponds, you know, it's not new, and it's quite a standard technique. It's just a lot more difficult in the open ocean, where you can imagine it's more challenging logistically to get out there, but it's also more challenging in that you're dealing with DNA that's in a huge volume of water, and that DNA is much more dilute. So in that sense, working in the open ocean in the high seas using eDNA monitoring is quite novel. And I did a lot of research as to what other people were doing to realize that we really wanted to work, like I say, with this lab that is they are the leaders in the field. And it just so happened that they happened to be developing this technique at the same time as we were thinking about using it. So it made quite a good collaboration, in that throughout the whole of the ship tour, we were essentially working with them. And as they were developing their methods, they were working with us to fine tune them. And so I think our work with the lab in the two labs in France, the University of Montpellier and SpyGen, really will help to develop this for more routine use in the open ocean. And to be able to go to, for instance, a seamount and spend, you know, a few days monitoring and collecting surface samples or samples at depth, you can imagine how much more easy that is than actually going and trying to video, or in some cases people may well have in the past had to actually physically catch the fish to try and identify them. I mean, like I say, it's not perfect because it is quite challenging. You're only getting a signal for presence of things that are sloughing a DNA into the water, but it is essentially quite novel and quite powerful. And I think it will become more and more powerful in the future when you're trying to assess biodiversity relatively rapidly in an area. And you can see from the results from Mount Vemma, you know, we would never, you know, when the team was there, they saw humpbacks, but they would never have seen necessarily a dwarf sperm whale, yet we were able to, you know, get a hit from the DNA. So it's really kind of interesting. And so there'll be a lot more work, I think, in the future, scientists work in the open ocean using this technique.
So interesting. And has there been something surprising from any of those legs that you've done, like the research that you've already analysed? Has there been a particular species that you found in the Antarctic, for instance, that you never thought that you would find in that region, or have you not had chance
No,
to?
we haven't had the data back from the Antarctic. So, I mean, there's a lot of things I'm interested in there. I mean, marine mammal wise, the number of different species in the Antarctic Peninsula isn't huge. It's quite sort of, in the summertime, the diversity is fairly low. There's a lot of humpbacks, a lot of, other species. But what I'm most interesting about those data, I think, are the ice fish, in that I know that the people have been working on ice fish right through the peninsula for quite a while. And so it'll be interesting to see whether, when we use when we look through our EDNA data, does that give us similar or different results from the sort of more traditional methods of monitoring? And for instance, we spent quite a lot of time at Elephant Island, and no one's characterized Elephant Island using EDNA. Does it tell us anything different from other sort of traditional surveys? So, I think I'm really interested. For me, it's the fish there, because essentially, it's a difficult place to work, and we managed to conduct these surveys at the surface without seeing a fish. We might have seen one, and so whatever we get from our EDNA data is going to be exciting, because it's not something that you can actually just go out and do quickly, like with a traditional method.
Yeah. Yeah, definitely.
I have a feeling we might have a lot of penguins in there too, though.
Yeah, I think you'll be right. Penguin soup.
Yeah.
So this is from Helen in Piha. I'm not sure if this is Helen that you know from your
time. It might be.
She has asked, did you see much tourist traffic while you were in the Antarctic?
Yeah, we did. And certain areas were really, really quiet. So our time in Elephant Island, we saw nobody. We saw one ship in the whole, I think, 5 days we were there. And that was, it's just such an incredible place because of its remoteness. And then as we traveled south to the more sort of well-trodden paths, it was actually kind of quite shocking in certain areas where it was quite small bays we were anchored in. And we were spending about 2.5-3 days doing our EDNA work, and we were photographing humpbacks, and then suddenly we'd turn around a corner and there would be a huge cruise ship that you can hear on the hydrophone chugging away, and they would have maybe 8 ribs cruising around looking at the penguins. And it's so stark to watch, because you don't expect it in a place like that. It's such a harsh and wild wilderness, and it's so incredibly beautiful. And then having a really bizarre looking giant mega ship turn around the corner, anchor, and then drop half a dozen ribs out that zoom around with lots of tourists all dressed in orange, was really quite shocking. And it did make you think that we ignore this sort of traffic and use of the environment at our peril, because we know that will have an impact on the different species that live there, and it is quite a special place. So it was really shocking.
Yeah, I definitely agree with that. Less of a sciencey question and more of a kind of personal one, I guess, next. What is your hope for the future of the oceans and places like the Antarctic?
Well, I think we have the ability now to be able to actually make some really good choices collectively as a species. Greenpeace is working on pushing forward a very strong ocean protection treaty, and I think that we're at a point where I sort of feel, in a way, quite excited that we can actually push this forward. And I think that for me, working with Greenpeace is a great privilege, in a sense, because as a scientist, you very often do a little piece of work, but it might not really be read by anybody else but other scientists. Whereas the work that we've been doing, the Poletopole ship tour, allows us to, give the whole of Greenpeace team some extra sort of extra weight to their message, essentially. And I think with the oceans at the moment, we are at this crossroads. We can make some good choices towards creating really widespread, well-connected networks of marine protected areas. And I don't see any barrier to that apart from just pushing it through policy. And that luckily enough, Greenpeace has a fantastic team to make that happen. So I think more and more we'll begin to see some of the oceans ecosystems recovering. You know, we've seen that with certain species recovering over time, and I think that, you know, I certainly have optimism in that sense, and I think we can see more of that. We just have to keep the pressure going. And so even though the UN meetings have been postponed, it doesn't stop our challenges, but it also gives me hope that we can make it happen.
Yeah, it's so true. And I think, I mean, I'm sure you saw it, Kirsten, there was a review in the journal Nature like earlier this year about ocean recovery being possible, and possible in the next 30 years,
Yeah.
which I thought was amazing, because I feel like all the stuff that I read is quite doom and gloom and quite like the oceans are going down and this is happening, and to actually read that it's still all possible if we actually make the right choices was really encouraging.
Yeah, I mean, it's quite optimistic to think that where we have made good choices about ecosystem management and the management really of human activities, where we have implemented that management, it does actually work. So we have proven examples now of how we can allow ecosystems to recover. We just have to make the right choices for the global oceans, essentially. And I guess that's what this whole campaign is about, is looking at it as on a much more global scale. And hopefully we'll be able to do that through the UN treaty.